So, before we start discussion on the reading, any questions about assignments or anything that have popped up since last class?
Yeah, I got one real quick.
Cool.
So, the ascocarps, when we pull those off, you said that we need to. Do we add water to that slide and then press down on it to?
Yeah, just a little bit. It helps with like light transferring through. So just like a tiny little drop, not even like a full drop, but just
Okay.
get some water on the tip of, I don't know, a knife or fork or something and kind of put it there. That helps the ascocarp stick to it, and it helps light get through the slide without refracting too much.
All right, thanks.
Mm-hmm.
I have a quick question too about the paper. So, or I guess the species, how many species there are. You want us to basically, because you set up to 500 words, so you want us to actually write, so you want us to read the papers, find the definition or what they base their argument on, and then explain a little bit?
Yeah, and
okay.
When I say read the papers, I don't mean, because you're probably going to have like 10 or 12 sources.
Lot.
I don't mean read the entire paper for detail, but more like the abstract. And if they are saying something useful, find that in the paper, pull out that quote, and then cite it. Yeah.
Okay, perfect. And that's enough. Okay, good. That's what I was. Okay,
that's enough.
Thank you.
Mm-hmm.
Yeah, I've got a question. Sorry if you've gotten this one a couple times, but so my bread mold, I was waiting to get some black mold to transfer over to the medium, but I've only had white mold the whole time, so I just transferred some of the white stuff over. Should that be okay?
Yeah, we'll see. If it starts making sporangia and doesn't have cell walls separating the sections of hyphae, then you've got a zygomycete.
Yeah, it's been a few weeks now, and it's just still all white, and so I just figured I'd transfer something over. So okay, thanks.
Now, if you're trying to get it to sporulate, how much has it grown in your petri dish? Has it kind of taken over the whole thing?
Yeah, I put it in a couple days ago, and it's already started growing quite a bit. So
it
hasn't really taken over yet, but it's getting there.
When it takes over pretty much the whole petri dish, pop it in the fridge. Just kind of see what happens. That may stress it out a bit and tell it.
Stress it out to start producing those spores.
Yeah.
Okay. Yeah, I'll do that. Thank you.
Mm-hmm.
Dr. Zahn, speaking of that, sorry, I have a quick question. So I decided to try, since my first experiment didn't work, I tried to do the smaller piece of bread and I got dirt from 2 sources and anyway, did that. And just to make sure, so it looks kind of like little black dots. Is that what I'm looking at? If they've actually...
Yeah.
Okay, good. So I got that. Okay, so I'll transfer it. And then can I transfer just that to the petri dish? I haven't transferred it yet because I was just letting it do its thing.
Yeah, just go in with a pair of tweezers and take out one of those black dots, and you'll be good to go.
Perfect. Okay, awesome.
Sorry, one more question about concerning that. So if we put it in the fridge and it's already taken over our petri dish, furthermore, are we looking for more of those little black dots, or?
Yeah, it's just kind of to confirm that you did get a zygomycete.
Okay.
So we're not doing this under sterile laboratory conditions, obviously, and that's okay, but it means we have to kind of do sanity checks on ourselves. So we picked up some black spots or whatever from our bread. We stick it on the dish. We want to see that it's the same thing. So we want it to look like it did on the bread, pretty much. So if you don't have any sporangia on your bread, you transfer over some white fuzz and it starts growing, takes over your petri dish for a couple of days, and then you stress it out. There's a good chance it's going to sporulate at that point. If not, then you just wait, right? Or pick up some of that fuzz, stick it on your slide, and look at it, and look for the septate hyphae. If there are no septi in the hyphae, then you've got a zygomycete, and so you're good to go. I just want you to have a zygomycete in culture. And
thanks.
Trying to confirm that in a number of ways in our limited kitchen mycology labs. Yeah.
So when we transfer over like one of the little black dots, are we trying to just get like the very end of it without any of like the hyphae or anything like that?
Ideally, the chances of you not getting hyphae are pretty slim.
But more or less trying to just get the little black dot.
Mm-hmm.
And we just need like one, is what you said?
Just one, yeah.
Okay.
Has anybody played around with their Sordaria crosses yet?
Is that the pollen one?
No, the pollen one is your chytrid.
Oh, right, sorry.
The Sordaria cross is the culture that I gave you that was already growing. It's the ascomycete, where we're looking for the paratecium and ascospores.
Oh, I've looked through it through a microscope. That was my question about earlier. It was if I need to put water on it. So I haven't done that just yet, but
Okay.
I've been able to take a couple pictures of just ascocarps before.
That's good. Yeah, you can even like set your microscope down on the auger dish itself and get some pictures of the ascocarp. But then to see inside, that's why we have to go to the slide. A little bit of water and kind of give it a light smash.
Okay.
To really spew out the ascospores.
Okay. Just for clarification, we're trying to grab the ones that are in between those two auger pieces, right?
Yeah, that's where we're going to have the best chance of seeing kind of that crossover effect between the light and the dark spores. Because remember, one of the bits of auger plugs that's on there is a mutant tan spore version, and the other is the wild type with black spores. When they meet up in the middle and do sex, then meiosis takes care with independent assortment and all that stuff. And not all the spores are going to be black, not all are going to be tan. So I want you to see kind of those differences.
Right. Okay, thanks.
Mm-hmm. All right.
Hey, I got a quick question.
Sure.
So, I was trying to take a look at the pollen tube for the chytrid, and I was having a pretty hard time actually getting some liquid out. It's like stuck at the bottom, and I didn't have a pipette or anything to get it out. Do you have any at-home suggestions for how to get that stuff out of there?
A Q-tip. If you rip off the cotton part, so you just have like a little piece of wood or plastic or whatever, that'll actually, some of that water will stick to that. The hydrostatic forces will kind of be overcome. You'll be able to tap that onto your slide.
Okay, cool. Thanks.
Yeah.
What if, can we just use like a toothpick? Would that be better?
Yeah, toothpick will work.
Okay.
Keep in mind that like you're sticking a toothpick in there, it's going to absorb some water instantly, and some of the pollen's going to stick to it. So you may have to kind of really rub it on your slide to get bits of pollen off.
Kind of scrape it off if we have to.
Yeah.
Okay.
And so just for clarification on the pollen, you're just looking for a little bit of a film kind of around the edges, and then as soon as you have that, if it's been like 3 days, you should be able to see one.
Yeah.
Okay.
Now I can't guarantee. These things never work out 100%. So there's going to be some subset of you that found a water source that has no chytrids in it. It's unlikely, but it's probably going to happen.
And are we trying to get the film off of that, or are we trying to get the pollen out of it?
You want the pollen out. The
Okay.
film itself is going to have chytrid hyphae in it, because these are like eucarpic chytrids. So they do make little rhizomorphs, and that's kind of what the film actually is. It's just those little rhizomorphs kind of growing around as feeding structures. But the body of the chytrid itself, where you're going to find the zoospores, that's actually going to be on the pollen grain. And so you want to be looking at pollen grains, and you'll see chytrids like latched onto them. If nothing else, send me, upload a photo of the pollen grain without chytrids.
All right,
You
thanks.
should be able to find chytrids, but at least send me a photo of the pollen showing no chytrids on it. If you're really lucky, you sit there looking at it for 5 or 10 minutes, the heat from the light can actually induce the chytrid to burst open and release its zoospores. And so you can see them swimming around.
Kind of general question. For chytrids, is there, as far as being found in water, any variation in like, is still water better versus running water or moving water?
The short answer is they're pretty much everywhere.
Okay.
Yeah. I've successfully used this pollen trap from all kinds of places, like frozen alpine lakes. You bust through the ice, pull out a little water. I found chytrids on that. A stagnant pool next to your house or on campus, I found them there. Found them in the Mississippi River. They're just kind of all over the place.
Wow. Okay.
Dr. Zahn?
Yes.
Did you say how many days we should be like incubating that water till we should be able to? I mean, can you do it right away, or is it?
3 days is usually enough.
All right, thank you.
So if it's been a few days, you may try scraping out some of that pollen, sticking it on a slide and taking a look. If you find pollen grains but no chytrids on them, wait a few more days, try it again. And if at that point you still have pollen grains but no chytrids, then you win, I don't know, buy a lottery ticket after that or something. Yeah. All right. Well, let's chat about ascomycetes and the reading then. If you have any other questions that come up, go ahead and shout those out too. But what came up for you in the reading? New things you learned, things you have questions about, any of that? We kind of covered a wide swath of ascomycota.
Okay. So can you explain the two types of, I think it's called enteroblastic, the phylloditic and enoloditic?
Yeah. Let me pull up the slide for that so I have a little visual aid for you.
Okay.
Or did I didn't put that on the slide, did I? That was in the reading.
Yes, that was in the reading.
Okay. There we go. Okay. So there are two ways of making a spore from a hypha, right? You can either have the hypha grow out and then kind of segregate itself and those pieces break off, okay? So that's called thallic. Thallic just refers to it's the whole body, okay? So figure C here, or part C of figure 8.5, is showing that it just laid down a cell wall and then that piece broke off, and we call that a spore because it's a asexual reproductive thing. Okay, so we call that a conidiospore or a mitospore or an asexual spore. The other way of doing it is to kind of squeeze it out of your top, kind of like a budding yeast, if you've seen video of that or looked at the resources. So it kind of buds out successively. Now, that's blastic. It's blasting out of the tip. Okay, blastic can be further reduced into a couple different varieties. If the entire cell wall goes along with it, like here in B, that's holoblastic. Okay, does that make sense?
Yes.
Okay. Now, over here in C, not the entire cell wall, but just parts of the cell wall, the inner structure of the cell wall go with it. So it kind of like breaks open the casing and then stuff squirts out. Okay. All right, so that's enteroblastic. The innards squirt out. The holoblastic, everything grows out of the tip.
Okay.
Does that help clear it up?
No, I was going down one more level. I was talking about it. It mentioned two types of enteroblastic, the phylloditic and analytic. I'm
Oh,
pronouncing those right.
yeah. Okay.
And yeah, right there.
Yeah. So it determines, what determines this is this kinidiogenous cell here. Okay. So whether there's a specialized cell that's doing the blasting or not, is kind of what determines whether it's phylloditic or analytic.
So if there is a special cell, it's phylloditic.
It's
and if
phyllodic,
there's
yeah. So
not.
I don't know that they've got a good picture. Yeah, this isn't bad. Okay. So phylloditic, enteroblastic. All right, we've got a specialized cell right here that's doing all of the blasting out, and it just keeps pumping them out from the base. So it grows from the bottom, if that makes sense.
Yes.
You know how to, okay, yeah. So it's kind of like blowing bubbles, and each successive bubble pushes the next ones off. In here, they break off one at a time. If it doesn't have that specialized cell, it just kind of keeps, it makes one, it flies off, and then it makes another, rather than continually pumping out a little stream of them. In practice, when you're looking at these under a scope, it's really tough to tell the difference of what's going on. They look the same on the structure, like on the conidium under a microscope. So I'm not too worried about you being able to like pick out the differences there.
Okay.
Yeah.
Hey, Dr. Zahn, can I ask a question about the quiz?
Yeah.
So question number 8. So I think I found it in the reading, but it took me a while to kind of like understand. So I'd love to just hear your words. It's what's the difference between the parathecium and the apothecium? Am I saying that right?
Apothecium?
Yeah, okay, there we go. So I found that in the reading, but it was just kind of like still a little fuzzy.
Okay. So it's the degree of which the fruiting body is closed around the mass of acai. All right. So in an apothecium, it's open up and the acai are just kind of exposed to the elements and they shoot their spores off into the world.
That's in an apothecium?
Yeah.
Okay.
I always think open. Apothecium. Let's see if I can pull up a quick photo for you. No, it's a drawing. Oh, my internet's having a grand old time. Okay. So here's a little cross section. So this looks like it's from a lichen. You can see this dark area here. Those are the acai with the ascospores inside, and they're just kind of open to the elements here. In a parathecium, these little lips basically almost close off to where there's just a little hole, and the acai gets squirted out through that hole, typically when it's disturbed.
Okay, that's what I was going to ask next. Is there a reason for either one to happen, or is it?
Maybe what is the function of an earlobe in a human?
Okay, yeah.
Okay,
gotcha.
This might be one of those like Stephen Jay Gould spandrels, right? It's something that evolved. It was good enough, and so natural selection didn't select against it. These things do get disturbed enough that they do get squirted out. Your Sordaria follows this parathecium pattern right here. So this is probably a picture of Sordaria that somebody's drawn. You can see it's just got a little opening. Okay.
Yeah.
if this gets disturbed, it gets stepped on by a bird or something, they just squirt out the top, like crushing a melon that has a hole in it. Yeah. These are also somewhat associated with fires. So in areas that have been recently burned, especially for really light fires, these can actually survive them, and that cracks them open and lets the spores out. So I don't know that there's a good reason. It does confer some extra protection on the ascospores while they're developing, but it's not like they can't get by without that protection, because apothecia are found all over nature also, and they're doing just fine.
So it's not like an apothecium, when it's put under stress, can become like a parathecium?
No,
it's just one or the other?
Yeah, or even a cleistothecium, if you remember that term, where it's completely closed and it actually has to be broken open.
Oh. Gotcha. All right, that makes more sense.
Mm-hmm.
Thank you.
The ancestral trait, looking at phylogenies, is an apothecium. That seems to be what parathecia and cleistothecia have evolved from. It's the apo version.
So maybe parathecium has some advantage.
Maybe. Sure.
Who knows?
Yeah, who knows? I'm not confident enough to say yes on that, because that kind of discounts all the things that do have apothecia. They're doing just fine.
Gotcha. Well, at least I better understand what they are now, so that's helpful. Okay. Thank you.
I see Hayden has her hand up.
Yeah. Can we go over verse 6 and 7? Sorry.
6 and 7.
Yeah, because I struggled to find the answers in the reading.
Yeah.
Is that the yeast one?
It's the one with like what the ascus is made of. I found it where diploid is made of the mother cell, right?
Yeah, I got that. And the haploid one, I was like,
I have no idea.
It seems like it's still made out of the mother cell, because there's not a whole lot else it could be made out of.
Did you know I wasn't the only one confused?
I've
Yeah.
already forgotten what questions I asked, so I have to look these up again. Okay. Oh. For the diploid and haploid Saccharomyces yeast, what does the ascus come from?
Yeah.
That? All right, so we can go right to the reading for that. And I believe it was...
Because it says it pretty clearly about the diploid, but it's pretty vague when it's talking about the haploid. It just says, oh, hey, these ascospores are inside the asci for haploid. And it doesn't say where the asci came from.
Mm-hmm.
All right. Do you see what I've done here?
Is it the same exact answer?
No. I tricked you.
It's a trick.
Where there's no, a haploid can't have asci?
Correct.
Oh, and I thought, oh.
Well, that's what I was
That's what
thinking.
I put. But I didn't put mother cell for the first one.
Yeah, so it's, if you look in the reading here, this kind of shows you. So here's the, the diploid version. We go through meiosis. The ascus is just the single cell of the yeast becomes the ascus. All right, so it is the mother cell. You can put that, or the entire body of the yeast. All right. What's different about these is like they're not multicellular, and so they don't have a separate ascus hanging out and a bunch of other cells surrounding it. It just becomes an ascus. But if you're haploid, you can't undergo meiosis. You can't make ascospores.
Okay.
So
So
I was
Ron?
That diagram then, because
I got you.
I was looking at it, I was like very confused based on the definitions they put at the bottom to explain the diagram. I thought that those were haploid cells.
Oh. Yeah, but they can't be, can they? No.
All right.
So I got you.
So scroll up just a tiny little bit, Dr. Zahn.
All right.
Because I was over this question. So in that first paragraph, 10.2.1, if you go like 3 sentences down, it says the life cycle of S is presented the haploid ascospores within the ascus.
Right.
So it's talking about what we're talking about in the question, the haploid in an ascus.
Well, that makes sense, right? Inside of an ascus is where meiosis happens, and so that's where you go from diploid to haploid. All ascospores in every species of ascomycete are haploid.
Oh, okay. So it's already after meiosis that they're talking about.
That's how you make an ascospore, is inside of an ascus, you go through meiosis. Yeah. So this is a semantic difference here that may have tripped you up. Ascospore versus ascus.
Yeah.
Did you know that every time, Hayden, you take a sip from that mug, it flips me off? That's incredible. Keep it going.
Not on purpose, I promise.
Okay.
I just like the mug.
Yeah.
I had a question about that one. Do haploid yeast cells, are those the ones that are like type A and type alpha that mate?
Yeah, so they're going to be either A or alpha. Yeah.
Okay, that's the haploid ones.
Yes.
Okay.
Then
Or
they
you
mate. That being an A can only mate with an alpha and vice versa.
Okay. And do yeast form conidiophores asexually?
Conidiophores. Okay, so the definition of a conidiophore is a structure that supports a conidium. And so I don't think I would. That may be a semantic thing. When a yeast is budding, that actually is a conidium, that new bud, right?
Oh, okay, that's what I was wondering. I guess
Yeah.
I used the wrong word there.
Okay. Yeah, and so it is a conidium, technically.
Okay.
The simplest, stupidest of all conidia, but technically it counts.
Okay.
And that refers to asexually produced spores, right?
Yeah. If they're sexually produced, they're ascospores in ascomycetes.
Dr. Zahn, can you explain a little bit the parasexual reproduction they go over? I think I understand it, but just be sure.
Okay, so this is where they're basically mating with themselves.
Okay.
So yeast can do this mating type switch since it's just a little locus on their chromosome. They can switch from A to alpha, right? And so some of them can actually essentially mate with themselves. They go through the motions of meiosis and everything. There's some reorganization, but there's no new genetic information brought in from like their sexual partner. So that's why they just call it parasexual.
Great. Thank you so much. I really appreciate that. Clarifies quite a bit.
Okay.
I have a question about that. So it says that, maybe I understood it wrong, but it talks about how it does it do crossing over during mitosis, or is it in meiosis? Because from what I previously understood, it's in okay.
In meiosis. Yeah.
Okay, thank you.
Yeah, because the book says, and I think that's why I was confused, because it talks about mitosis and kind of how it like goes from diploid to haploid in mitosis.
Yeah, by losing chromosomes somehow?
Yeah.
What was going on with that?
Wait, where is that?
Let's see, I have the page number. I'll give you 1 second. It's 8.33. It's page 230. And actually says meiosis does not occur, and instead haploidization takes place by the successive loss of chromosomes.
I totally missed that. like forever.
Maybe I read it wrong,
Just
but
like
I thought it
15
did say that.
Yeah, I
thought it
was super
said
confused
crossing
about
over in mitosis as well.
this
which I was confused
parasexual
about.
section.
Yeah.
Wow. I've read this chapter like 4 times. I've been a mycologist for 15 years, and I had no idea. That's embarrassing. Yeah, that's actually new to me. Okay, so they're just dumping half their chromosomes, essentially.
Yeah.
What's the benefit of that? Like, why would they do that?
Hang on. I'm still trying to wrap my head around this. I'm gonna have to get back with you on it. I have no idea. I'm gonna have to make a call to somebody, a fungal geneticist, and ask them about this. I'll throw it out on Twitter today and hopefully have an answer next week for you.
Awesome. Thank you.
Mm-hmm.
So it's safe to say that won't be on the test.
Oh yeah. And if it had been on the test, I would have gotten the wrong answer in there. So that's cool.
Okay, non-real.
Yeehaw. All right.
Hey, Dr. Zahn.
Yeah.
So you talked about how haploid alpha can only mate with haploid A, but I thought I read somewhere about like only heterozygous diploid that had been formed from alpha and A could go on to form asci, but it talked about like homozygous diploid as well.
So yeah, but they're still gonna have. They're still. That's not in the yeast necessarily. That cleared up. It doesn't.
I'm still a little confused because it seemed like it was.
All right. So
it seemed like
A and
homozygous
alpha
diploid came from alpha or AA.
Yeah, A and alpha is a mating system found in yeast and some other fungi. All yeast have it, but there are other mating systems. So there are like, what's the term? Tetrapolar, where you have basically 2 loci, and both of them have to be different in your sexual partner. So you've got an A and alpha, and then like a G and gamma, and both of them have to be different in order to mate. Okay, you've got fungi that can mate with themselves and don't have mating type issues, and you've got some weird fungi in the basidiomycetes that have 30,000 different mating types that we know of, probably more. So the A and alpha, don't think that applies to all ascomycetes. Really, we're just talking about saccharomycetes there.
Okay. And then for the saccharomycetes, when you asked the question about the haploid, if for that we put that the haploids, 2 haploid of opposite mating type would have to mate to form a diploid, which could then become an asci, would that be an acceptable answer?
Yeah, because that's not wrong.
Okay.
Yeah.
Cool. Thanks.
Mm-hmm. All right. Any other things? Bring them up. Let's see if you can embarrass me again.
Well, this one's probably pretty easy, but
He says knowingly.
the 3, as I'm asking about it, the 3 growth forms seen in ascomycota.
Yes.
Yeast, filamentous, and dimorphic.
Okay. I wasn't sure if that's what you're referring to. It's very
Yeah.
for some reason.
I
I
put
got
that
that
down.
wrong. No, I
and
was
then
like
just in-
budding, fission, and
Well, budding and fission work for yeasts, so that's one growth form you got.
Could we also go over like the Warrenin bodies? I don't know if that's how you pronounce it, real fast, and just like kind of how they function.
Okay, the Warrenin bodies, they, as far as we know, are kind of like a blood clotting mechanism almost, I guess you could think of it. So when the hypha gets damaged, they're hanging out next to the little pores inside by the septa, and they plug up the hole.
Okay, so it's kind of like the same way like when we cut our finger, like the blood cells like move there and like kind of clog it. So it's like the same kind of idea.
Yeah, so they're there to kind of
Okay.
like plug up the hole and clot it, stop things
Okay.
from rushing out. And part of that's because of their shape, but really they like chemically bind, right, protein interactions to that and like plug it up basically permanently.
Okay, thank you.
Hmm.
So
that
in the
a-
reading,
oh,
it talks
sorry.
about how they will like congregate in areas where it's damaged.
Yeah.
So is like the hole that the pore that they're plugging considered damage, or is it that they specifically are just trying to plug any hole?
They're hanging out like ready to plug a hole at any point. It's like platelets in your blood. Okay. If there is damage, they do kind of get pulled out because like all the liquid from the cell is rushing out. It got broken open. They get pulled in. They kind of plug it up at that point.
And is
so,
there any sort of like evolutionary advantage that for that pore being there? Like,
why is the pore there?
yeah.
The pore is there so that nutrients can still get passed back and forth, but it's small enough that nuclei can't get through. And so this is kind of handy because you can still get nutrients, you know, water, salts, all kinds of stuff. Mitochondria can move back and forth, but every segment of that hypha is going to have either one or two nuclei. And so this prevents a section of hypha from having like 16 nuclei in it, like we get in the zygomycetes.
So is it constantly plugging that hole, or is it moving to allow more flow?
It's actually just kind of floating around until there's damage, and then it's like a little airlock, right? Everything starts flowing out. It gets sucked into the pore, and then like binds to it to shut down things from moving past it.
Okay.
Yeah.
Is the Warrenin body just in the ascomycetes, or is it in all septate fungi?
As far as we know, just in ascomycetes.
Oh, okay, cool.
Yeah. Which seems weird because basidiomycetes are quote unquote more highly evolved, but they don't have those.
Wow.
So they
So
lost.
if there's no nuclei sharing, where does the second nuclei come from sometimes? That would be an ascomycete cell.
Right. So when they're dikaryotic, meaning they got both parent nuclei hanging out in the same cell, they want one from one parent, one from the other, hanging out in the same cell. They don't want anyone else around. Okay, no other nuclei there. You're asking like, how do you get two nuclei into the new growing?
Yeah, did they just force them in and then put a septa behind it?
Yeah, so when let's see if I can draw this. All right, so we've got our growing hypha. It's got two of each nuclei in the cells. when it's time to grow through mitosis, they just both split through mitosis, and one of each version moves into the new cell that's growing. I don't know if that is helping or making things worse.
Just kind of looks like they move from the one to the other in your drawing.
Yeah, so they split via mitosis, both of those nuclei. And
so they duplicate. Okay.
Yeah, they duplicate. And then you've got spindle fibers and stuff pulling the new nuclei on ahead.
Towards a pole.
And then a cell wall comes down, shutting them off from each other.
Okay, got it.
Yeah. Now, when we look at, let me see if I can find sexual. When they're making asci, yeah, here we go. All right, you see this figure?
Yes, with the shepherd's crook.
Yeah, okay. So you can see that they're splitting via mitosis, and they're getting pulled into this new penultimate cell here. Okay, so that's kind of what's going on. This is the ascomycete as it's making asci, ensuring that each cell has one of each version of the nucleus in there. All right, and so it just goes along making a bunch of successive little hooks. So this folds over, they both split. We lay down some cell walls to make this new cell that will become the ascus. Okay, and so here you can see it undergoes karyogamy in here, then meiosis, and then a second round of mitosis. And all the while, this next little cell is growing out, hooking over, segregating off the next pair. That's going to become the ascus.
And they
So
only showed 2 of them, but it's just continuously doing that, right?
Yeah. And so that's why you get an apothecium with like 10,000 asci in it, because it's just constantly doing this little loopy loop. Every time it loops back around on itself, it shuts off that becomes an ascus, and it loops again, that becomes a new ascus. Try as I might, I cannot find a video of this anywhere. I know I've seen one, because that's how I first learned how to understand this, was actually seeing like a little stop motion video of this happening. So maybe give it a shot on YouTube or something. I just could not find it.
So 8.6.1 print pretty in depth on this formation of the ascospores, and they really hit heavy on like the formation of some membranes uniting around them. Is that super important to know? Because it just kind of seems like, hey, they get individualized.
Yeah, I think that's probably the take-home message there. If you knew anything about the authors of this book, you would know why they harped on this, because that's their area of study. And so there you have it. This textbook kind of goes a bit heavy into the like cell biology of fungi, I think, overlooking a lot of things. But other than that, it's a good book, especially for overview of our different groups.
Yeah, I noticed parts that really went into the cell biology and all that, and just throwing tons of that at you. On those parts, can we kind of skim over those?
Yeah, I think so. I mean, this is not a fungal cell biology class. We're trying to get like an overview of fungal groups, some of the weird things they do, learn a few of the terms. I wouldn't go too deep into that. if you remember from like the Chytrid chapter, I think they spent 2 or 3 pages talking about how the substructures and proteins of the flagella were arranged.
Oh yeah.
Yeah, so
Okay.
when you're reading these, I would read it rather quickly. Then look at the main points from the website that I want you to take home and go back through looking for those. Don't spend too much time memorizing the structure of chitin or anything like that. Or I should say, don't spend any time memorizing the structure of chitin. Okay. What about our 12, 17, the pyrenomycetes? Any of these special groups get you tripped up?
Is that why I see Justin walking up and down there? Huh? Is that why I see Justin walking up and down?
What's that? If that was a question, I couldn't hear it. I guess that was not a question. Nothing else? This is your chance to get your participation points.
I mean, there's no question about the rest of the chapter, but I did just
Yeah, it doesn't have to be about the chapter. That's fine.
As a safety backup, I was worried about my first bread mold not really growing, so I took from a different water source up in Provo Canyon, threw it on there, and it's growing exactly the same with the white filaments not turning black as well.
Mm-hmm.
So I did just start. I just took the sporangia from one of them and threw it in there, in one of the Petri dishes. But
the sporangium you took, did it have like a spore mass on the top, a little ball or something, or was it just a thread?
Yes, it had a little ball.
Okay. Yeah, then you're good.
Okay, but what are the odds of me getting 2 that aren't black? That's what we're looking for. Like, am I just getting unlucky?
Yeah, I think you're getting kind of lucky. You said you took it from a water source, not from soil?
So I took my initial one from my front yard, from the soil, mixed some water in there, dirt, whatever.
Right.
And then for the second one, I took some water from a mountain stream. I used that for the Chytrid pollen, and then I also mixed a little bit of dirt in it from right next to it and put it on a piece of bread as well.
Okay. The odds aren't super slim. I would say you probably did get a bit lucky not getting Rhizopus twice. Rhizopus is the black bread mold that always happens to your bread. It happens to your strawberries in the fridge, all that stuff. It's one of the most abundant common fungi on Earth, probably. It's soil dwelling. It's just kind of everywhere, and its spore masses are black. So there's a chance that you got something else that outcompeted it, or you just didn't have Rhizopus a couple times. I wouldn't get freaked out by that necessarily.
Okay, but the point is we're just trying to get some sporangium.
Yeah, sporangium, any zygomycete.
Got it, thanks.
My expectation was that probably everyone would get Rhizopus, because it's so common. But obviously that's not working out, and that's fine.
I've got a random question about Saccharomyces.
Yeah.
You said that it formed that white coating on the outside of grapes.
Yes.
Are there other substances that make up that coating, or is that just purely Saccharomyces yeast?
That's Saccharomyces. Yep.
And do you know of any other fruits that it forms on?
It's on all fruits, but it just like turns up on grapes. They tend to have some tannins and stuff that make them unwelcome for a lot of microbes. But if you were to take a strawberry or a peach, you'd get Saccharomyces from that also. Anyone have a plum tree and know that white stuff that you can kind of rub off of your plum? That's Saccharomyces also. Like even at the grocery store, you go buy a plum, it's kind of got that just a white powdery looking film on it that you can rub off. Okay.
Thank you.
I got a question about the quiz on number 10. It talks about the, how do I say this? Deuteromycotini?
Deuterocotina. Yeah.
Thank you. That one. So is the reason why they don't it's no longer in use, is that because of like they've never formally established any classifications on it? Sorry. Is that the reason, or is there more to it?
I was hoping somebody would ask me about Deuteromycotina. It's not a real group. Everything that we couldn't find a sexual stage for got thrown in there. Everything. Okay. And so this is before we really knew that it's before we had phylogenetics and we could place these things in the tree. There are even basidiomycetes in the quote-unquote group Deuteromycotina. Sometimes these are called the fungi imperfecti. They're not perfect because they can't do meiosis and have sex. Okay, so anything that was like trapped in an asexual cycle, or we thought was, we just threw it into that group. And so you'll see, if you go to the library at UVU, there's like 2 or 3 books on the taxonomy of the Deuteromycotina, but none of it's real.
So everything that was put in there, you said they no longer use that term anymore. So have they been moved out to other areas because they've been classified now, or are they still unclassified?
Well, we kind of figured that they were all ascomycetes, and we just figured, oh, these don't do sex anymore, and so that's a trait that they all have in common. And so we thought, okay, this must be a group within ascomycetes, and we'll call them the fungi imperfecti or Deuteromycotina. And so we thought it was more or less a monophyletic group within ascomycetes of just things that could no longer do sex. And now that we can sequence them, we see that they're spread out all over the place in Ascomycota and Basidiomycota. And so we just threw out that group now that we know where they belong. And so sex has been lost a lot of times in fungi, not just once.
Okay. Thanks for clearing that up.
Yeah.
So was everything that was categorized in that, was that basically dispersed out into different groups and that group name was collapsed? Or just that whole thing in general was...
So in Deuteromycotina, there were a bunch of different ascomycetes and even some basidiomycetes. We now stick them where they belong, and since there is no grouping that's monophyletic of fungi that don't have sex, we just discard that name completely. There is no group called Deuteromycotina.
Okay, that makes sense.
Yeah.
Hey, Dr. Zahn.
Mm-hmm.
I've got a question on I'm forgetting if it was number like 6 or something, but it was the one when we had to look up what was the sexual form of the fungus, I believe.
Oh, yeah.
So I assume that probably should have been easy, but when I looked it up on what is it called, WebBank or whatever it's called, I feel like I couldn't get. Yeah, I feel like I couldn't get a very straightforward answer, so I was just trying to find answers wherever I could find them, and I can't remember what I put. So you have to look at mine, but it was like I saw online, and it was like the first word, instead of putting the asparagus, it was like empercillium or something like that, and that was considered the sexual form. But then when I submitted the test, it was quite different. So I was wondering maybe what would be the proper way to go about that.
All right, so I'm going to walk you through it here. I'm just going to copy Aspergillus acutus. I'm going to go here to search, basic search on names, taxon name. Just going to pop that in there and hit enter. All right, so here's Aspergillus acutus. Zoom on down here. Teleomorph synonym. There it is.
Okay, so I was on this page. How would we know that that was the sexual version?
Because teleomorph means sexual, anamorph means asexual.
Well, that's it right there then. Okay, thank you.
Yeah.
Yeah, it took me a while to figure that out. At first I was like, do I need to like look in this article? And like, but yeah, I had the same issue.
So I put down that name, but I also added Blazer at the end of it because I wasn't sure what like I was supposed to add in, and it was wrong. Will I still get points for that or not?
Oh, yeah. Okay. Yeah, I'll add points. If you put Blazer, that's just the name of the person who identified this and stuck it in that genus. So
I
that's
just wasn't sure what that was, and so I added it anyways. But yeah.
Gotcha. Yep. Who was that? I'll need to make a note.
Leah.
Oh, okay. Okay.
So I've got one more question on the Saccharomyces.
Okay.
So it says that there's like 10 to 16 species of Saccharomyces, and then it says that there's 25 strains. Are the different strains just essentially, like any other strains of like a virus or something? Or something its genetic makeup has just changed slightly
But they can still actually mate with each other. Yep.
Oh, okay.
Yeah.
That's what I was wondering is if they could kind of intermingle and if that made it.
And that's pretty out of date. I'd say there are many hundreds or thousands of strains of even a single species. Saccharomyces cerevisiae, the brewer beer yeast and bread yeast. Let's see if I can find. Not too long ago, they did a huge phylogenetic analysis genome-wide of just baking yeast, essentially, and there must have been 600 different strains on there, and they group out really well according to like location. Okay, so if you go to Germany, you're going to have this monophyletic group of Saccharomyces yeasts, which is different from Belgium and all that. It's just because humans have been intervening so much in the evolution of Saccharomyces species. Which makes sense. It's one of the most important microbes we have, like commercially and industrially. So we've selectively bred strains of it that can tolerate alcohol concentrations of up to like 30% for making biofuels. Okay. We've got strains that make San Francisco sourdough taste like San Francisco sourdough. A bunch of good stuff. Okay. Any other lingering questions?
I have a question.
Okay.
With the find a chytrid, would it be appropriate to use water from a freshwater fish tank?
Yeah.
Okay. What about a saltwater fish tank?
Probably not.
Okay. I was just curious.
Yeah, probably not.
And Dr. Sun, you said later on we'll go over pathogens for humans, right? Because I mean, there are a bunch in ascomycetes.
There are a bunch in ascomycetes and in zygomycetes, not so much in basidiomycetes, but yeah, we've got a whole
So we'll cover those later in the semester.
just on medical mycology. Yeah.
Because that'll be really interesting.
And I can't say that I'm going to be going into too great of depth with it. I'm not a full mycologist, but you'll probably get about as much as you will in med school, unfortunately.
That's fun.
So one day.
Yeah.
Amazing.
One day. Mm-hmm.
I have a funny thing to say.
Okay.
It's not about fungus, but I don't know if you know who Walton Goggins is.
Mm-mm.
Anyway, I'll send you a picture of him. I think you look so much like him. It's an actor. Anyway, it's not a bad thing. It's just funny.
I had a question. Who sent me that? I think it was Chandler. Put a question up on our teams about diploid versus haploid in yeast. Was that you, Chandler, with us? No. Okay. At any rate, down here in our resources, there is a paper about that very thing. Okay, so if she's watching this later, definitely go to that. Any questions about the videos, Neurosporic Crosser or Beetle and Tatum? Are you guys even clicking on these resources? I guess I should put a tracker on here and see, like, get IP addresses for who's doing that.
I watch them. I just don't have any questions. We're good.
Okay.
Yeah, same.
All right.
Hey, Dr. Zahn.
Yes.
Just a quick question, just because I'm always like looking forward to tests and stuff. So for the test, is it just basically going to be like a giant quiz with 3 times as much stuff, or is it going to be quite different? Because I don't know about, it seems like everyone else has a lot better grasp on fungus than I do. Because doing these quizzes, honestly, for me, I struggle. And so I was just wondering, like, what should I be trying to prepare for to, I don't know, get a better grasp on this information?
So on the website, I'll just keep harping on this because it's going to remain true. Click on the major topics for each week. This is what I'm testing on. These
Okay.
things here. Yeah. I hope that helps.
So is the format the same as your quizzes, though?
Yeah, it's just going to be a big Canvas thing.
Right. Answer the questions like.
Yeah. Give your best answer, a few multiple choice, some true false.
Okay.
For like, I'm not going to rule out having you do something like the ascomycete assignment, where I have you find a paper or two covering some topic and summarize them, or look up the teleomorph synonym or the anamorph synonym for a given fungal species. And then when we get into the end of the class and we're learning about kind of the fungal genetics, I may give you a DNA sequence and say, what fungus is this? Okay. So there may be some outside of Canvas stuff also.
And the tests are going to be open note, right?
Open note, open internet. If you want to get a study group together and take it all together, I don't care, right? I'm looking for you going through the motions and like learning the material well enough to Google it well. If that helps.
Which is applicable in the real world.
Yeah, I'm not interested in you memorizing all this stuff. As you can see, I just embarrassed myself really thoroughly giving you the wrong definition of parasexual. So glass houses, I can't throw stones. All right. So being able to be familiar enough with the terms that you know how to Google about mycology, I think is the most important part. In the process, you're going to get knowledge, right? If somebody says that's a basidiomycete, you're going to have some kind of epiphany in your brain somewhere in there that goes, oh, I know what those are. Okay. But no, I don't have everything memorized about them. That's okay. All right. So focus on these major topics for the research or for your exams on each of those. Our first exam is actually coming up in 2 weeks. I'm going to post it up on Canvas. Try to have it there by like Tuesday afternoon in week 5, and it'll be due Friday night.
So sorry, just real quick, how long do we get to take the test? Is it just on Friday, or is it like, is there a couple days?
Tuesday through Friday.
Okay, sorry, I did.
Oh yeah.
Just caught that.
And I know you said it won't have a time limit. Will it just be open? So once we start it, can we, if we, I mean, I guess what I'm wondering is if we don't finish all right then, can we like save it and come back to it?
Yeah, so Canvas is actually nice about that. If I don't put a time limit on the quizzes, you can stop it and come back the next day and just pick up where you left off as long as you save your progress. Yeah.
Should we be watching out for tricky questions like there's been on the quiz?
Oh,
I see a little smirk going on there.
I may try to get you. I may ask about zoospores and ascomycetes, and you should have enough wherewithal to say no. Right. Yeah. So it's possible.
Is the same true for the quizzes? Are we able to stop and come back to them? Because it took me
Yeah.
a while to do it, so I don't know if I need to do it all in one sitting or not.
Yeah, you can stop and come back to them.
How do you save them? Because I was worried that if I closed it, it was just going to turn in what I had.
No, if you close it, it should be fine. You can come back to it. Even if it logs you out, you can still come back in and it'll say resume quiz on it.
Yeah, just don't click submit.
You don't have to save it, yeah.
Okay.
You just type in the answer and it saves it for you.
Cool.
I had a question about the final real quick. I was just noticing on Canvas that it says, what does it say? It has a little exclamation point right next to it, and it says that this assignment does not count toward the final grade.
Oh, that's just me accidentally not unchecking that. That's the default for Canvas.
So you don't want to be nice and just let us not have to take the final, huh?
Well, that's no fun. I mean, the final, it's not going to be like super difficult or anything. I'm just kind of going to put together some of this stuff, some big idea questions about fungi, give you some DNA sequences, find out what these are, that kind of thing.
Right. I just noticed that. I just thought I'd bring it to your attention.
I'll fix it. But yeah, it'll count towards your grade.
Okay.
Yeah. All right. Well, I think we're good. Just keep working on those assignments. Let's see here. Go ahead and get started on your reading. Once again, there's no paper for the basidiomycetes because there's plenty enough reading already about them in your textbook. So we can go ahead and get started on that, and I will see you next week.
Hey, real quick.
Yeah.
When are you going to have the quiz up for the next week's reading?
This weekend.
Okay.
Yep.
All right, thanks.
Yeah.
Doctor's on.
Mm-hmm. Yes.
Oh, I was just saying thank you and have a great.
All I heard was Dr. Zahn. Oh,
Oh,
yeah.
no, I said thank you, Dr. Oh, I probably didn't click the mic on time.
Gotcha. All right. We'll see you next time.
Sounds good. Have a good weekend.
Stay safe out there.